Hi, Jeremy Cordeaux in the Court of Public Opinion. I'm just on air here to let you know that we'll be live streaming the Court of Public Opinion every Friday between 9 o'clock and 12 on jeremycordeaux.com. Please join us. We'd love to have you.
Dr. Travis Brown, what is This Medical Life all about?
This is the pursuit of knowledge as we learn about diseases from the ancient times to the present day. These are the stories of medicine.
Dr. Travis Brown, just recently I introduced my daughters to a viral YouTube sensation called Double Rainbow. It's hilarious. If you ever want a moment of ridiculousness, it's it. But we're not talking about that. It just reminded me because we're talking about the double helix in this episode.
We are talking about the double helix. So this is a story that is well known in the medical community and the scientific community at large. It even goes into popular culture about the discovery of the DNA double helix. And the structure is important here that our guest will go into a little bit later. But this is one of the monumental discoveries of modern medicine, of science, and most of our audience will be very familiar with this story. And there are, if you look at this story, you can see characters and you can see ambition, you can see betrayal and tragedy. And if it sounds like a novel, that's actually by design. And the reason we sort of look at this is because it has all the elements that you kind of expect to see in a novel.
That may be the case, but I'm sure if we dug hard enough, just about every major breakthrough in science and medicine probably has an intricate web of story.
It does, and this is why this podcast works. If you actually dig into the details of the story, you can actually find some rich characters and also ambition, people, scientific discoveries, and it's not pretty, to be honest, and that is in itself compelling. And look, nucleic acid was discovered in the late 1800s. So the ATCG building blocks of life were known, but it wasn't known how it worked. And the discovery of actually it containing genetic information was done in 1943 by Oswald Avery. And he showed that a gene, so to speak, can be transferred from one bacterium, it was a pneumococcus, to another bacterium. And you could actually transfer that information from one to the other. And that showed that information is in this, quote, gene. And so he was looking at this, but no one knew how that gene worked. And so in the 1950s, we have two groups that are the closest to the discovery at this time. And one was the University of Cambridge, and this was Jim Watson and Francis Crick. Now, they are described, so to speak, as Jim Watson was young, brash, and obsessive. He is the person who actually wrote the novel later on in this story, so to speak.
So I'd play him in the movie.
You would, young, brash, zealot. And so then we've got Francis Crick. Now, he is a brilliant free spirit, an inquisitive mind, but easily distracted. And
Travis Brown.
And then we have the other group. This was King's College London. And this was a person, Maurice Wilkins. Now, he was a brilliant nuclear physicist who had been disillusioned by the creation of the atomic bomb.
Oh, wow.
And what he turned to then was he turned to biophysics. And he started looking into this X-ray crystallography, which is effectively an imaging technique using X-rays of molecules. And so this gets down to tiny...
That's mind-boggling.
Exactly. And this is the tiny, minute areas, and they're imaging it. Now, he was working with his mentor at the time, John T. Randall. Now, Maurice Wilkins was described as quiet and non-confrontational. And John T. Randall hired the new up-and-coming. Now, her name was Rosalind Franklin. Now, she was a young woman and a brilliant scientist and an expert in this X-ray crystallography. but she was described at the time
Yes.
as forceful, brisk, as confrontational. And so you have Wilkins, who's there, who believes she was employed to work under him, and she believed she was to work independently. And so immediately you have, now, excluding the story, it's widely known that they did not get along. And so I suspect that's probably John T. Randall's fault that he didn't communicate that very well to his team at that time. And so when we're looking at this, we get into the 1952, and we have Watson and Crick who propose a DNA structure, and it's completely wrong. They have DNA that has 3 strands, it's inside out, and this is seen by Franklin and dismissed immediately. So she said they're wrong. And because of this, as well as some of the other problems as seeing as the Cambridge people were stepping on the King's College toes, the actual project Watson and Crick were working on was shut down.
Oh, wow.
And so the reason it started up again was because there was a group out of the US that started competing and being interested in the DNA structure, and the University of Cambridge didn't want that to happen. So then they started up again, and Watson and Crick were able to progress their studies, so to speak. But in 1953, Watson visited Wilkins at King's College, and Wilkins, apparently unbeknownst to Franklin at the time, showed Watson this classic photograph 51 or exposure 51, and Watson described the moment.
The instant I saw the picture, my mouth fell open and my pulse began to race.
Now, apparently, they understood the helix structure of the DNA, but they didn't know the distances. And so a report by Franklin to her boss was passed on to their boss, who shared it with Watson and Crick. And from that information, they were able to work out the distances. And so when they were able to work out the distances through calculations, they then had made the discovery of the structure of the DNA helix. And this was the discovery of the century. And later, they ended up presenting that information. Watson and Crick presented it, and there's that classic picture of them looking at each other with the DNA helix. Yes, it's widely publicized. And the model presented of the double helix strand. And Wilkins and Franklin were consulted on this, and they agreed. They said, yes, this is correct.
But it was their work.
Well, that's the strange thing. Not only that, it was agreed that Watson and Crick would be the sole publishers of it, and that Franklin and Wilkins would then publish supporting data later to confirm the findings.
This is like the sport of cycling.
And it's just jaw-dropping, but at the same point in time, there was some celebrations done by the university a few months later, and everyone invited, but Franklin didn't attend. And she was busy moving on and went to her new area of study. And so when we look at all of this, it's hard to sort of piece it together. And look, what ended up happening 5 years later in 1958, Rosalind Franklin died of ovarian cancer. She was 37 at the time. And so if I can just take a moment just to look at how remarkable her career was. She was known as an expert in her early 30s in a male-dominated field in the 1950s. And so if you can think about her when she's described as brash or confrontational, she'd almost have to be. Like in that kind of a field, the kind of barriers that would have been there for her and put in her way would have...
And you've got the double standard too. She was just acting how all the blokes around her would have been acting. But of course, women can't act like that.
And it's just an amazing sort of turn of events. And then in 1962, Watson, Crick, and Wilkins were then given the Nobel Prize for physiology or medicine. Now, there are a few kinks in this, meaning that the Nobel Prize is notably not given to people who have died, so
That's right.
posthumously. but it's one of those things that, was she excluded? Was this discovery sort of taken her out of that? And here's the thing. Why does this all sound compelling? Why does this all sound like a novel? Is because we've got the mix of facts and fiction in here. And this is primarily done by Jim Watson, who in 1968 wrote the novel, The Double Helix, a personal account of the discovery of the structure of DNA. And so it is a molding of, is he framing it? Is this the politician's autobiography?
I'm sure it's factual. I'm sure it's 100% under the microscope objective.
And so what we've come, and why we're discussing this now, is because there's been some evidence that have come to light. Our expert is coming up to discuss, well, maybe it wasn't as one-sided as it seems to be at this point in time. So wanted to dig this story out, and I wanted to talk to our guest coming up about the information that hopefully shows a little bit more that Rosalind came to the forefront.
Now, I was about to show you a theory that I've got, but I'm going to keep it under wraps just in case you go and take the glory for it. Nathaniel Comfort is Professor of the History of Medicine at John Hopkins University. His interests lie in the histories of genetics, eugenics, genomics, and biomedicine, as well as bioethics. He's the author of The Tangled Field, Barbara McClintock's search for the patterns of genetic control, and The Science of Human Perfection, how genes became the heart of American medicine, and he's editor of and contributor to the Pandas Black Box, opening up the intelligent design controversy. Back in 2015-2016, he was the Blumberg Professor of Astrobiology at the Library of Congress, NASA. Comfort's scholarship has been covered by news outlets worldwide, and he's written for The Atlantic, The Nation, Nature, Science, the New York Times Book Review, and elsewhere, and has appeared on PBS, National Public Radio, and the BBC. He's working on a biography of the biologist James D. Watson as well, just in his spare time. Today, Professor Comfort is our guest on This Medical Life podcast. Welcome.
Thank you. It's nice to be here.
We're so looking forward to this, and I'd like to start with the obvious and the simple question, perhaps. How does the story of the DNA double helix discovery actually start?
Well, simple question, obvious question, yes. Simple, no. Actually, it depends on your perspective. The usual way that the story is told is it begins with Watson arriving at Cambridge University in England, where he soon, on a postdoctoral fellowship, where he soon meets Francis Crick, who is a superannuated graduate student. He's 12 years older than Watson, but still working on his PhD. Watson is a postdoc and only 23 years old. So they're kind of an odd couple right from the start. And they find themselves office mates in the Cavendish Laboratory of Physics, a renowned laboratory of physics research. And as Watson tells the story, he went there to find the secret of life. And he found that Crick was also interested in finding the secret of life. And so they sat down and spent the next year and a half madly pursuing both it and girls and tennis balls and, having all sorts of fun. And by the way, making the, arguably the most important discovery in biology in the 20th century. That's the way that it's usually told.
Oh, but there's a but coming, isn't there?
There are a lot of buts coming, but that's Watson's version of the story. And the whole story of the double helix, this has been written about many times by journalists, historians, scientists, you name it. It's received a lot of attention over the years. And it's very complicated because Watson published the first account of the discovery, and he did so in a very strange way. the book really cannot be taken at face value as literal history. Watson didn't intend it to be taken, or at least not 100% intend it to be taken that way. It's fictionalized. It's based on a true story, but he made it into this kind of epic coming of age slash eureka story that was designed to attract young people into science. He wanted to tell an exciting story. And so he sort of goosed the real story in certain ways to make it exciting for young people. And people have taken it as the literal, sincere truth, and it isn't.
So then, now that's the double helix. He called it was a personal account of the discovery of the structure of DNA, and in that he has some characters which are based on reality, as you said. There is himself. There is Watson and Crick, himself and his co-partner, but then there's also Rosalind Franklin. Now, how does she fit into the traditional story of this DNA double helix discovery?
And by the traditional story, you mean Watson's account?
That's right.
Right. In
there is a distinction.
There is a distinction. In Watson's account, she is this terrible Rosie who is ill-tempered and is supposed to be Morris Wilkins's assistant. Wilkins eventually shared the Nobel Prize with Watson and Crick. He worked not at Cambridge but at King's College London, and Franklin is in the lab there with Wilkins. And in Watson's story, she's supposed to be his assistant, but she refuses to think of herself this way, and she is dowdy and grumpy and always trying to get. And she is a good crystallographer, but she can't understand her own data. And a lot of the story is built on, she adds a lot of dramatic tension to the story by being this sort of sinister figure. And he gives a very, very unflattering portrait of her that, again, some people have said wasn't that far wrong, but they never say that on the record. And it was, I mean, his portrait of her draws on every sexist stereotype you can imagine of the dowdy woman scientist, right? And so, rightfully so, many people have been quite upset about Watson's treatment of Franklin in this book.
Now, there is the mention of the classic photograph 51. Can you tell us about that?
Photograph 51 was an X-ray diffraction picture. What they're trying to do in the King's College London lab, what they're trying to do is understand the structure of the DNA molecule by crystallizing it and then putting it in front of an X-ray beam and putting behind it a photographic plate. They shoot an X-ray beam at the molecule, and as it hits the beam is tiny. It's on the order of the size of the atoms, and so when it hits the atoms in the molecule, it scatters. Okay, and it leaves a scattering pattern on the photographic plate behind it, and from that scattering pattern, it's laborious but possible to back calculate the structure that produced that pattern. Okay, that's X-ray diffraction or X-ray crystallography. And that's what Franklin was an absolute wizard at. And photograph 51 is one of those photographs of the DNA molecule in one of its forms called the B form. Wilkins discovered that there were two forms, A and B, and Franklin worked on the A and B forms and found that she could translate one into the other just by raising and lowering the humidity in the chamber where the sample is changed. So photograph 51 is a photograph of the B form of DNA, and it's the more humid version. And this is the one that is more biological. The dried out version, the A form, gave more detailed pictures but less lifelike, because the inside of a cell is a very wet place, right? The humidity is very high inside a cell. And so this is the photograph that in Watson's story becomes the climax moment. Wilkins has been given photograph 51 by this graduate student, Raymond Gosling, who worked with both him and with Franklin at different times. And Gosling gave him the photograph, Wilkins, and Wilkins showed the photograph to Watson. And in Watson's story, he sees this and his jaw drops open and his pulse begins to race. And he realizes immediately the secret of life, right? Franklin had taken the photograph 10 months earlier, in May of 1952. This is not a new photograph. It is not something that they had never seen before. There were plenty of images of the bee form floating around, and had been for a long time. There had been photographs of the bee form taken before Watson even got to Cambridge, right? The photo 51 is a particularly pretty and clean image, but it's not actually that significant. That's what we found. But Watson turns it into the climax moment of the story, and that's when he can, he sees the light, all the scales drop from his eyes, and he understands the secret of life, right? He's by seeing that this is a helix. And there's a lot of mythology wrapped up in that.
Before you continue, is there some P.T. Barnum-like characteristics inside this character of Watson?
Some P.T. Barnum, that's interesting. Yeah, you could say that. I think you could say that. I've never thought of him as a P.T. Barnum character, but yeah, there's some salesmanship. There's definitely a lot of showmanship. Yes, he is telling a goosed up, sensationalized version of what happened, and he's not being very clear about when he's being historical and when he's being novelistic.
Yeah.
Right. So people, and then other things, later we can talk about later if you like, but other things happen to encourage the misunderstanding. And Watson did nothing to dispel that misunderstanding.
You never let that get in the way of a good story.
Is it?
That's right.
Can I ask? Is it actually written as almost a detective novel that is gripping and or is it sort of almost fan fiction of himself going about from the 1960s?
Kind of, yeah. It's a complicated book and it has many layers, but that's not completely wrong. No, it is sort of fan fiction. It's self-mythologization is what it is, right? He's creating the myth of DNA and making it coextensive with his own biography. Okay? And the Double Helix was the most successful, the best-selling version of that. But he created that mythology in several different ways and pulled them together that made himself really the founding figure of molecular biology and of DNA, the genome. And that's not to say he wasn't important in that work. He certainly has had a remarkable career in science, and also a notorious one in more recent years that maybe we can talk about later. But he's underestimated as a storyteller.
All right, there's so much I want to get into. Shall we spend a couple of moments just on Crick, though? Because he's also an interesting critter, and I believe likes to finish other people's crosswords.
It's been said, yes. My co-author in the Nature article on Franklin that came out a couple of weeks ago is Matthew Cobb of Manchester University, and he's working on a book, a biography of Crick, while I'm working on a biography of Watson. And we're going sort of side by side. We exchange many emails a day and share our findings and ask each other questions, and it's tremendous fun. And he finds Crick to be a very lively character. He was, I mentioned that he was older than Watson. He was still a graduate student, although in his mid-30s, substantially because of World War II. He had been doing war research for the Admiralty on physics-related topics, and he said that he was interested in biology all along, but his early training was in physics, and he was extremely bright, garrulous, and very outgoing. And he had this constant stream of ideas. He was a magnetic person to be around. People just were enchanted by him because he would come in and dominate the conversation with all sorts of interesting ideas. And yes, the bit about solving other people's crosswords, he did like to come in and say, "Oh, hi, what are you doing?" And then you'd explain what you were doing, and then he would solve it for you, like right there. And so people started to become a little bit wary of telling Francis what they were working on, because like they wanted to find the answer. And he was so quick, he not rarely could just like see the answer. He was a real ladies' man. He had an open marriage, and was constantly had various sorts of lady friends and so on. He was a very kind of ribald and witty and colorful character.
So they both seem that both Watson and Crick seem to have that flamboyance, if you like, as part of their own DNA, ironically. Let's take a pause and come back in a moment, because we want to dive deeper into the meat of this story. Unfortunately, at the age of 37, Rosalind Franklin dies of ovarian cancer in 1958. In 1962, Nobel Prize in Physiology or Medicine was awarded to James Watson, Francis Crick and Maurice Wilkins for the discovery of the molecular structure of DNA. Wilkins, with his colleague Rosalind Franklin at that time, provided the X-ray diffraction patterns that Watson and Crick used to discover the DNA helix. However, Rosalind was not named or awarded posthumously anything. A common perception of this is that Watson and Crick essentially stole Franklin's work and took credit for her work. However, recent evidence has challenged the traditional view and suggests that Rosalind Franklin understood what photograph 51 represented, and she was an equal contributor to solving the DNA helix structure. Now, Professor Comfort with Matthew Cobb from the University of Manchester are doing research into James Watson and Francis Crick respectively. They studied Rosalind's notes and found a draft news article destined for Time magazine and a letter from one of Franklin's colleagues to Crick. Now, Professor Comfort, how has this shed light on the DNA double helix story?
Well, it doesn't alter the fundamental history as historians have known it. It changes the tenor or the mood of the story in pretty important ways. What we find is historians have known that Franklin had contributed a lot more and have understood better why there was so much tension between Franklin and Wilkins. And Watson, in portraying Franklin, really used Wilkins's point of view. He was channeling Wilkins's view of Franklin in his portrayal, which was basically all he knew of Franklin at the time. So that's all he got about Franklin came through Wilkins, who was constantly complaining about her. And so there was a book in 1975 published in response, almost as a rebuttal to Watson's book, The Double Helix, by Anne Sayre, called Rosalind Franklin and DNA, in which she goes through the double helix almost chapter by chapter and rebuts the things that Watson says. Okay. And she mounted a important defense against Watson's sexist account. Okay. I think the book is a bit unfortunate in the certain aspects of the writing, but it had an important function. It challenged Watson's narrative, and it kind of opened up the scholarship and suggested that there were other interpretations possible. So Matthew and I went in to visit the Rosalind Franklin papers when we were both in Cambridge. We thought, okay, as we should make the pilgrimage and go see her papers. We'd seen what most of them are online, so it was mostly a sort of symbolic gesture, and historians like to get their hands on the actual documents, But as we were going through this stuff, things started to click in us. We realized the problems with even the reasoned historical accounts. That, first of all, looking at her notes, her research notes, particularly for this colloquium in 1951, right at the beginning of Watson's time in Cambridge. And he'd only been there a few weeks, and he attended the seminar that Franklin gave. And we have Franklin's detailed notes for that seminar. Scholars have looked at these notes before, but we looked at them, and we think we saw them in a new way. The way she was describing her work suggested that she was absolutely justified in taking the approach she did. So even the people who are defenders of Franklin tend to adopt a kind of version of Watson's account by trying to explain why she didn't get the double helix and how it was she missed this really important clue and so forth. And we realized that she didn't miss much. She had a very detailed understanding of the structure of DNA in 1951, before Watson had even started working on it. So we read through the notes for her colloquium, and we realized that her emphasis on the A form rather than the B form was perfectly justified given her approach and given her background and her training. It made sense for her to, as a chemist trained in X-ray crystallography, it made perfect sense for her to focus on the A form. This was not some gigantic mistake that she made. The reason is this: the A form is much more detailed and much sharper. What you do, remember those photographs that I mentioned that had the little spots exposed on the photographic plate? Each one of those spots or lines or various shapes is a data point. And what you do is you measure those and you count them and you look at the angles and the relationships between them. So the more spots you have, the more data you have. The more data you have, presumably the more precise structure you can devise from it, right? And she's a crystallographer. She called the A form the crystalline form. If you're a crystallographer, what you want is the crystalline form.
Yes.
The B form was she called the paracrystalline form, near crystalline, but not quite. And we read her description of the B form. She's in the seminar. She's got her notes, and she's going through her data. She's comparing the A and the B forms, and she describes the A form and says that it's a helix and has various parameters. The B form, she writes, when it has too much water, it gets distorted, distended, and the thing basically falls apart. So that told us, other people have had that text to look at, but it hasn't struck anyone before that, okay, what she's saying is that the B form is a mess, right? It looks to her, as a trained crystallographer, it looks like a big watery mess where the molecule is starting, the water molecules are getting in there so much that they're twisting and distorting the structure, and basically it's starting to dissolve in water. That's how she looked at it. And that was a perfectly reasonable interpretation, right? It's only, and this is what's so hard about this, is getting past our present day understanding of what DNA is and means, and trying to reconstruct what it's like when you don't know the double helix yet, right? If you don't know what it is, it's not obvious at all. And that's what Matthew and I are trying to do with this. So that was one thing that we found in the archives. The other was the letter that you mentioned, or the magazine article. You probably have seen the photograph, the famous photograph of Watson and Crick in front of the big DNA model. Crick is pointing at the model with a slide rule, and Watson is sitting beneath him, kind of gaping up at him, in the sort of idolizing way, right?
Yes.
So we found, so that has been known to have been part of a series of photographs taken by Anthony Barrington Brown in 1953 in May, shortly after the Double Helix papers came out. It was known that he was sent there by Time magazine to take pictures to accompany an article, and it was known that the article was never published. And that was it. That was all. And those photographs had been studied a fair amount. So we found the article.
Oh.
Okay. We found the article, and we were flipping through a folder that was really thin and easily overlooked. But as we're flipping through it, I noticed an envelope with the Time magazine logo on it. And I said, hey, wait a second, Time magazine. And we looked at what followed, and there it was, an article on the Double Helix intended for Time magazine by an author named Joan Bruce, a writer in London. We haven't been able to find out much about Joan Bruce yet, but we do know that she wrote an article on the Double Helix story just after it had been published. So this is the first narrative account of the Double Helix. And she tells it very differently from Watson.
Oh.
She tells it very differently. She tells it as a story of two teams of scientists, one at Cambridge, one in London. The London team is doing the experimental work. The Cambridge team is doing the theoretical work, and they are working together, checking each other's data, providing ideas, and not always finding that the other group was 100% accurate. They're making corrections and so forth. So this is a dramatically different account in tone and tenor from Watson's rip-roaring tale of deviousness and cunning and single heroes and so forth, right? And dowdy women who get in the way of the bright naughty boys who solve the big problems. Okay, here we have Franklin, first of all, given equal weight to Wilkins as a member of as one of these two dyads. Right, that's the key to her picture of DNA, is those two dyads of people, and that's split along theoretical and experimental lines. Yeah, and Franklin is given equal standing in relation to Wilkins, and the Cambridge, the King's College team is given equal standing compared to the Cavendish team, who are Watson and Crick. So it makes a symmetrical account. That is, we found really quite striking. Then recall that we found this article in the Rosalind Franklin papers at Churchill College, Cambridge. Joan Bruce had sent the article to Rosalind Franklin for asking for corrections and to make sure she got the science right. She didn't get the science right. Scientifically, it's kind of a disaster. She had some major misconceptions about how biology works, right? And we think that that's probably why the article was never published. If somebody had had the patience to fix the science and keep her interpretation of the social relationships, that she got right, right? The way we would understand DNA very differently today, I think. This certainly could have done, right? And so she sent it to Franklin, and apparently, we don't know this for sure, but apparently only sent it to Franklin. We've looked in the Watson, Crick, and Wilkins papers, and there's nothing even analogous to this. Yeah. And it's okay. They might have gotten it and thrown it away. Watson didn't throw much away. especially about the double helix. So, his papers are enormous. Wilkins saved everything. He was kind of obsessive. So,
And Crick would have finished off the article for her.
Yeah, right. Exactly right. Exactly right. So it reads to me like Joan Bruce is taking a gently feminist stance by showing the article to Franklin, getting her feedback on it by portraying her very much as one of the equal players, and not in any sense distorting the picture, but taking a bit of a stand, right? It reads that way to me, that in the tone. And so that was really remarkable to us. And that kind of put us in a new mindset that like shifted the axes for Matthew and me. And we started playing out that scenario. Okay, if it was like that, what does that mean? Well, that would explain this and that and that and that. That seems pretty right, right? And it sheds light on Watson's construction of the story 15 years later as well. And you can sort of see now more clearly what's mythologized and what was closer to history and so forth. So it was quite an exciting thing to find. So Nobel Prizes by strong convention are never given posthumously. So having died tragically young of ovarian cancer, probably due to all those x-rays that she worked with, without, people often were pretty cavalier about radiation in those days. And it's been suggested that that's what happened. So she died 4 years before the Nobel was awarded to Watson, Crick, and Wilkins, and 2 years before they were even been nominated. The first nomination that came in for them was 1960. So it really was too early, and that's a shame. But there's another piece of the story, which is that Franklin's seems to have considered her most important work to be what she did after the double helix. If you look at her tombstone, where she's buried, Matthew has been there and sent me pictures. I have not been there, but her obituary in the New York Times when she died, she was a 37-year-old British scientist who got an obituary in the New York Times. So she was not unknown. She also got a substantial obituary in Nature. She was a highly respected scientist, but what was considered her main work was that of her work on virus structure. And that seems to be what she identified as her most important contribution, and as evidenced by the fact that the initials DNA do not appear on her tombstone, but RNA does, and virus structure does. And also it seems to have been her reputation, regardless of independently of what she thought of herself, it seems to have been her reputation, was primarily as a highly skilled researcher on the structure of viruses.
Wow. And we could have done with her again recently during the COVID-19 pandemic, couldn't we?
Indeed.
Yes. We've talked about some of the history around the people of this story, but why was the discovery of the DNA double helix so significant?
Well, the first thing that we have to clarify is that the significance isn't in the helix, it's in the double. Okay. A lot of structures in nature tend to grow in a helical way. It's a very common form just by the nature of things like cell division and gravitational forces and atomic forces and so on. And there are other molecules that take a helical form. Watson has emphasized the helix because it's a shape that everybody can visualize. It becomes a great logo or icon or emblem, and it's instantly recognizable. But the spiral itself isn't scientifically interesting. What's interesting is that there are two chains winding around each other in this helical shape. Okay. And the chains have a backbone made of molecules called phosphate groups, and a base which is a molecule that has, in DNA, it has 4 different forms. And people may know the initials that they're known by, A, C, T, and G. And what's significant about it is these form a like a spiral staircase, like a ladder, right? A spiral ladder up a lighthouse. And the treads of the staircase are where the bases join. So the 2 strands join in the middle of the tread of each rung of the ladder, and they pair only in one way each. There are 4 types. A always pairs with T, and C always pairs with G. And that means, first of all, that that a strand of DNA a single strand can have any sequence of bases on it. It's not determined, and so there's nothing structural about that. No matter what sequence you have, the double helix has the same form, and that means that it's possible to contain the genetic information to specify proteins, right? Because it's indeterminate. It can be anything, and Franklin realized this, and the fact that you have these 2 pairing mechanisms means that one strand is an indeterminate sequence of bases in one direction, and the other one is an exact match in the other direction.
These are the missing pieces for all the genetics episodes that we've had thus far in our life. Look, you've really whetted our appetites to actually read your writing. It's been quite illuminating in this episode, but coming to a close now, and I'm just wondering if there's any final thoughts you have regarding our understanding of the DNA helix controversy, and anything lingering that we can take away from this story.
Well, we can take away from this story the importance of getting the history right, that it matters that we understand what really happened as opposed to the sensational stories, that doing so is important for recognizing the unrecognized voices in science, issues of women in science and other underrepresented and underprivileged groups in science. And I think most of all, in my mind, it illustrates how difficult it really is to think historically. It's so difficult. Matthew and I have run into this many times ourselves, and we're constantly getting over this feeling of thinking we know how the story is going to turn out, right? And the difficult thing is trying to put yourself in the mindset of being Rosalind Franklin or Jim Watson or Francis Crick or Maurice Wilkins, not knowing what seems so obvious to us now. It's so simple and elegant that it's almost impossible to forget that you know that, and to look at the world the way it looked to those people who figured it out for the first time.
Wow. On that note, Professor Nathaniel Comfort, thank you so much for joining This Medical Life.
Thank you. It's really been a pleasure.
This Medical Life is recorded in the Talked About Marketing Studios in Adelaide. For show notes and more information about the podcast, visit thismedicallife.com.au. You can contact the hosts via Twitter. Dr. Travis Brown is at Dr. Travis Brown. That's DR for doctor. And Steve Davis is at Steve Davis. Editing and production is by Tim Whiffen. Design is by Tom Buzenjud. This has been a PathNotes Proprietary Limited production.
Hi, Jeremy Cordeaux in the Court of Public Opinion. I'm just on air here to let you know that we'll be live streaming the Court of Public Opinion every Friday between 9 o'clock and 12 on jeremycordeaux.com. Please join us. We'd love to have you.